Hello and welcome to this tutorial on stages of drug development. We will discuss how development of new drug usually takes place and what are the challenges we face. Number one, the development of new drug requires a lot of pain and effort as the process of new drug development is expensive and time consuming. It requires a number of chemical, pharmacological, and toxicological research. Critics claim that the cost of development and marketing are grossly inflated by marketing activities, advertising, and other promotional efforts, which may consume as much as 25% or more of the company's budget. There is an interesting article on how to slash the cost of drug development. The economists at Tough Center for the Study of Drug Development in Boston have collected data from pharmaceutical companies and use it to calculate the average cost of developing a new drug. The most recent estimate is $1.4 billion. Even the chief executive of London-based pharmaceutical giant GlaxoSmithKline, Andrew Whitty, has called billion-dollar estimates. He attributed a huge sum of spending too much time on failures. Then, the drug candidate can be killed as a result of safety concerns, poor efficacy, or profitability worries. And he argued that the companies could save money by dropping bad leads. Others say that the figure is inflated by large and excessive trials done to prove that a new drug works just slightly better than the existing one. You can read this paper, the link has been given to you. Now, next question is why drug development requires so much time? The answer is very simple. You need to understand the whole process of drug development. Human clinical trials have been divided into four phases: phase 1, phase 2, phase 3, and phase 4. Phase 1 study understand the safety of the drug that could be done on 20 to 100 subjects. Phase 2 will be studied to understand the effectiveness of the drug that the trial will be done on 100 to 200 patients. Whereas phase 3 will be conducted under randomized double-blind clinical trials for larger population, 1,000 to 6,000 patients. Whereas phase 4 is the post-marketing surveillance trial are conducted after a drug or device has been approved for consumer sale. Pharmaceutical companies have several objectives at this stage. Number one, to compare a drug with other drug already in the market. Number two, to monitor the drug long-term effectiveness and impact on the patient quality of life. Number three, to determine the cost effectiveness of the drug therapy relative to the other traditional and new therapies. For example, Alzheimer's disease treatment, there are 105 agents under pipeline divided into phase 1, phase 2, and phase 3. The trial include a wide range of clinical trial on population, understanding the mechanism of action, and require a substantial number of clinical trial participants. Then, what are the approaches that help to discover a new drug? Screening of biological activities of large number of natural products, bank of previously discovered chemical entities, or large libraries of peptide, nucleic acid, or other organic molecules. For example, morphine was first extracted from opium for pure form in early 19th century. It was widely used for painkiller during the American Civil War, and many soldiers became addicted after that. Codeine was used after that because it is less powerful and has been synthesized and isolated by French scientist Jean-Pierre Robiquet in 1830 to replace the raw opium for medicinal purpose. Second, the chemical modification of known active molecules known as analogs, for example, antimalarial drugs such as chloroquine and amodiaquine. Number third is identification or elucidation of new target. For example, thienopyrimidine derivative exhibit as identical as a new drug target against Mycobacterium tuberculosis. Which is number four, the rational design of new molecule based on the understanding of biological mechanism and drug receptor structure. For example, metal-based complexes that work as anti-cancer for inducing apoptosis. Next is drug screening, which involve a variety of assays at the molecular, cellular, organ system, and the whole animal level to define the pharmacological profile. For example, anti-infective drugs to test against a variety of infectious organisms, some of which are resistant to standard agents. What will be the key observation during screening? The molecules is also studied for a broad array of actions to determine the mechanism of action and selectivity of the drug that will help to identify the expected and unexpected toxicities. But what is the secret to design a good drug? A good predicted clinical model is effective to design a good drug. The whole animal study are generally necessary to determine the effect of the drug on an organ system and disease model. Effect on the cellular function to determine the type of drug, such as agonist, a partial agonist, inverse agonist, or antagonist at relevant receptors. These are concepts we will discuss later on. Isolated tissue would be used to characterize the pharmacological activities and selectivity of a new compound in comparison with the reference compound. For example, the antihypertensive drug, animals with hypertension would be treated whether to see the blood pressure was lowered in a dose-related manner and to characterize the other effects of a compound. These studies might suggest the need of the further chemical modification to achieve more desirable pharmacokinetics or pharmacodynamic properties. We will discuss what are the pharmacokinetics and pharmacodynamics in the later on lectures. Then we have preclinical safety and toxicity testing that consists of different type of tests such as acute toxicity, subacute toxicity, chronic toxicity, effect on the reproductive performance, carcinogenic potential, mutagenic potential. And all the approaches would be different. For example, for acute testing, we will use only 2 species but 2 routes for small time. Whereas for chronic testing will be done on rodents and 1 non-rodent species for 6 months. If we compare the carcinogenic potential, it will be a long-term research for 2 years, and we will discuss about the prolonged effect of the drug use, and we discuss the determine the gross and histological pathology. So if you see all these testing, these 6 testings are all different and depending upon the use that is required. So thank you.